Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on biological membranes. Let's go ahead and get started with problem number 1. Problem number 1 says a student is trying to determine the type of membrane transport occurring in a cell. The student finds that the molecule to be transported is very large and polar, and when transported across the membrane, no energy is required. Which of the following is the most likely mechanism of transport? A says active transport, B says simple diffusion, C says facilitated diffusion, and D says exocytosis. The problem says the molecule is very large and polar. That's important because the cell membrane is made up of phospholipid bilayer with a hydrophobic interior. Large or polar molecules, they can't just slip through on their own. They need some kind of help to cross. The question also says no energy is required. This tells us that this is not active transport because active transport always requires energy input, usually in the form of ATP or an ion gradient. If no energy is used, then we're talking about a passive process. So that narrows it down to simple diffusion or facilitated diffusion. Simple diffusion works only for small nonpolar molecules like oxygen or carbon dioxide that can pass directly through the membrane. But our molecule, it's large and polar, so it can't use simple diffusion. That leaves facilitated diffusion. In facilitated diffusion, molecules move down their concentration gradient with the help of a transport protein or channel, but without any energy input. And this matches the situation perfectly. Large, polar, no energy needed. The correct answer for problem 1 is C, facilitated diffusion. Problem 2 says, a researcher treats a solution containing animal cells with Ouabain, a poisonous substance that interferes with the sodium-potassium ATPase embedded in the cell membrane, and the cell lyses as a result. Which of the following statements best describes Ouabain's effects? A says treatment with this results in high levels of extracellular calcium. B says treatment with Ouabain results in high levels of extracellular potassium and sodium. C says treatment with Ouabain increases intracellular concentrations of sodium. And D says treatment with this decreases intracellular concentrations of sodium. The sodium-potassium ATPase, this is an active transport pump in the plasma membrane that uses ATP to move sodium and potassium against their concentration gradients. Specifically, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell with each cycle. And this maintains low sodium and high potassium inside the cell. That's really important for osmotic balance and cell volume. Ouabain inhibits this pump. If the pump is blocked, then sodium can no longer be effectively transported out of the cell. So as a result, sodium is going to begin to accumulate inside because sodium is still entering from passive leak channels, but it's not being pumped back out. So over time, this increase in intracellular sodium, that's going to disrupt osmotic balance, and it's going to cause water to enter the cell and lead to lysis. So the best answer is that Ouabain increases intracellular concentrations of sodium, and that matches answer choice C. Problem 3 says, resting membrane potential depends on blank. Statement 1 says, the differential distribution of ions across the membrane. 2 says active transport processes. And 3 says selective permeability of the phospholipid bilayer. Okay, so let's recall what we learned in lecture about the factors that establish and maintain the resting membrane potential. First, we discussed that there is a differential distribution of ions across the membrane. Sodium is more concentrated outside the cell, while potassium is more concentrated inside. Second, we covered that active transport, mainly the sodium-potassium ATPase, that's essential for maintaining these gradients over time by pumping sodium out and potassium in against their concentration gradients using ATP. Third, we emphasized selective permeability of the phospholipid bilayer, which is more permeable to potassium than sodium at rest. And because potassium can diffuse out more easily and sodium's movement is more restricted, a negative membrane potential is established inside the cell. So all three of these factors, ion distribution, active transport, and selective permeability, they work together to produce and sustain the resting membrane potential. So the correct answer is going to be D, since all three statements are true. Problem 4 says, which of the following is not a function of the cell membrane? A says cytoskeletal attachment, B says protein synthesis, C says transport regulation, and D says second messenger reservoir. Now let's think through the main functions of the cell membrane. One key role is cytoskeletal attachment. The membrane can anchor elements of the cytoskeleton, and that's going to help maintain cell shape, and it's going to facilitate structural changes. Another major role is transport regulation, since the membrane controls the movement of substances into and out of the cell through things like channels, carriers, and pumps. The membrane, it can also act as a second messenger reservoir with certain lipids and proteins in the membrane serving as sources or anchors for signaling molecules. However, protein synthesis is not a function of the cell membrane. Protein synthesis takes place primarily in the ribosomes, which may be free in the cytosol or bound to the rough endoplasmic reticulum, but not in the plasma membrane itself. Therefore, the correct answer for problem 4 is B. Problem 5 says, the dynamic properties of molecules in the cell membrane are most rapid in blank. A says phospholipids moving within the plane of the membrane. B says phospholipids moving between the plane of the membrane. C says proteins moving within the plane of the membrane. And D says proteins exiting the cell through exocytosis. We know from lecture that molecules in the cell membrane can move in different ways. Phospholipids can move laterally, meaning they shift positions side to side within the same layer of the bilayer. And this type of movement is very common and extremely rapid. It can occur thousands of times per second. This lateral movement is a key reason why the membrane is described as fluid in the fluid mosaic model. Another possibility is for a phospholipid to move from one layer of the bilayer to the other, and this is a process called flip-flop. This is much slower because the hydrophilic head group would need to pass through the hydrophobic interior of the membrane, and that is energetically unfavorable. Now, there are special enzymes called flipases that can help, but it still doesn't occur as quickly as lateral movement. Proteins can also move within the plane of the membrane, but they are much larger than phospholipids, and they're often anchored to the cytoskeleton or to the extracellular structures, which is going to limit their mobility. Finally, the option describing proteins exiting the cell through exocytosis. This is a separate process involving vesicle fusion with the plasma membrane. It's a bit unrelated to the question. So, since we said lateral movement of phospholipids within the same plane of the membrane is by far the fastest motion, we can confidently say that the correct answer to problem 5 is A. Problem 6 says, which lipid type is least likely to contribute to membrane fluidity? Now, in lecture, we discussed that phospholipids can have saturated or unsaturated fatty acid tails. Saturated tails are straight and they can pack tightly together, and that makes the membrane less fluid. Unsaturated tails, they have double bonds that introduce kinks. That's going to prevent tight packing and increase fluidity. However, not all double bonds behave the same. We specifically learned that cis double bonds create these kinks, but trans double bonds do not. Trans bonds keep the fatty acid tail straight, kind of similar to a saturated tail. And because of that, transglycerophospholipids pack more tightly and they reduce membrane fluidity. So now looking at the choices, we have unsaturated glycerophospholipids. When this doesn't clarify cis or trans, it usually means cis because that is more common. So this has cis double bonds that increases fluidity, so not the answer we're looking for. B says transglycerophospholipids, straight tails. This is going to be tightly packed. That's going to decrease fluidity. This is our likely answer, but let's still work through C and D. C says cholesterol. Now this can both increase and decrease fluidity depending on temperature. And since temperature is not clarified here, it's not a confident answer, especially not compared to B. D says unsaturated sphingolipids. Again, it doesn't clarify cis or trans. That means it's cis. So this contains cis double bonds that would increase fluidity, not what we're looking for. So the correct answer is going to be B, transglycerophospholipids, because their structure most reduces membrane fluidity. Let's move on to problem 7. Problem 7 says a membrane receptor is most likely to be a blank. A says embedded protein with catalytic activity. B says transmembrane protein with sequestration activity. C says membrane-associated protein with sequestration activity. And D says transmembrane protein with catalytic activity. Now, receptors are proteins that detect and respond to specific molecules, typically located on the cell surface so that they can interact with extracellular signals. For them to transmit information from the outside to the inside of the cell, they need to span the membrane. That's why most receptors are transmembrane proteins. Now, some transmembrane receptors also have intrinsic catalytic activity on their intracellular portion. And a good example is receptor tyrosine kinases, which span the membrane, and then upon ligand binding, they're going to activate an enzymatic function inside the cell to start a signaling cascade. Now, that matches one of our answer choices perfectly here, which is D, transmembrane protein with catalytic activity. Problem 8 says, which of the following is not a cell-to-cell junction in animals? This is a very straightforward question, and the correct answer is C, plasmodesmata. These are plant cell-to-cell junctions that allow cytoplasmic exchange between neighboring plant cells through their cell walls. All the other 3 options are cell-to-cell junctions found in animals. So 8 is C. Let's move on to problem 9. 9 says, which of the following is true of diffusion and osmosis? A says diffusion and osmosis rely on the concentration gradient of only the compound of interest. B says diffusion and osmosis rely on the concentration gradient of all compounds in a cell. C says diffusion and osmosis will proceed in the same direction if there is only one solute. And D says diffusion and osmosis cannot occur simultaneously. Diffusion is the movement of a solute down its own concentration gradient. Osmosis is the movement of water through a semi-permeable membrane in response to a solute gradient. But the thing that actually moves is water, so it follows its own driving force. In both cases, we track the gradient of the substance that's moving. That's why the correct statement is that diffusion and osmosis rely on the concentration gradient of only the compound of interest, and that's answer choice A. Let's also talk about why the other answers are incorrect. B says they rely on the gradient of all compounds in a cell. This is not how we model transport. Each species moves according to its own gradient. C claims that diffusion and osmosis will always go in the same direction if there is one solute. That's not necessarily true. And D says they cannot occur simultaneously. They can occur simultaneously. It's not an all or nothing kind of situation, however. So they can sometimes occur simultaneously. They can sometimes not occur simultaneously. So B and C and D, they're not correct because the statements are a bit variable. So that means 9 is A. Problem 10 says the bulk movement of liquid into a cell through vesicular enfoldings is known as blank. A says phagocytosis, B says pinocytosis, C says exocytosis, and D says drinking. Now, the bulk movement of liquid into a cell through these enfoldings, this is a form of endocytosis, where the cell membrane folds inward to enclose material in a vesicle. The key here is that the material being taken in is liquid, not solid particles. So that specific process where liquid is being taken up, not solid, that's called pinocytosis, often nicknamed cell drinking. Now, in pinocytosis, the cell takes in extracellular fluid and any dissolved solutes by forming small vesicles. The correct answer here is going to be B. Problem 11 says, which of the following is least likely to be the resting membrane potential of a cell? A says negative 70 millivolts, B says negative 55 millivolts, C says 0 millivolts, and D says positive 35 millivolts. Cells almost always maintain a non-zero resting membrane potential, typically a negative value, because this electrical difference across the membrane is essential for responsiveness to stimuli. The resting potential, it stores electrochemical gradients that ion channels and signaling molecules rely on to function. If the potential were 0 millivolts, however, there would be no separation of charge, meaning no stored gradients for later activity. This would make it impossible for the cell to generate action potentials or to perform normal signaling. So the other answer choices, they're valid because they can represent values that occur during resting or active states of the membrane. But 0 millivolts is not consistent with a functional resting cell. That's why the correct answer for 11 is C. Problem 12 says, how does the inner mitochondrial membrane differ from the outer mitochondrial membrane? A says the inner is more permeable and lacks cholesterol. B says the inner is less permeable and lacks cholesterol. C says the inner is more permeable and has cholesterol. And D says the inner is less permeable and has cholesterol. Now, the inner mitochondrial membrane, it's highly specialized because it houses the electron transport chain and the machinery for ATP production. To keep these processes efficient, the inner membrane is much less permeable than the outer membrane so that most ions and molecules can't just freely pass through it. This is going to allow the mitochondria to maintain a strong proton gradient, which is essential for making ATP. Another key point is that the inner membrane lacks cholesterol, which is different from many other membranes in the cell. So putting it all together, the correct answer here is going to be B. The inner membrane is less permeable and it lacks cholesterol. Problem 13 says for most cells, the extracellular calcium concentration is around 10,000 times higher than the intracellular calcium concentration. What is the membrane potential established by this electrochemical gradient? To figure out the membrane potential from this gradient, we can use the Nernst equation. At physiological temperature, the Nernst equation can be simplified to the potential is equal to 61.5 divided by the charge of the ion multiplied by the log of the concentration of the ion on the outside over the concentration of the ion on the inside. Now, our ion of interest here is calcium, and calcium has a charge of +2, so that is our Z value. The concentration ratio, that's going to be 10,000 to 1, and that's because we're told that the extracellular calcium concentration is around 10,000 times higher than the intracellular calcium concentration. So this part of our equation is equal to 10,000 divided by 1. This is just equal to 10,000. We want to write this in scientific notation because it's going to make the log calculation without a calculator a lot easier. This is just 10 to the power 4. Now we have everything we need to plug things into our equation. We have 61.5 divided by 2 multiplied by the log of 10 to the power 4. Now I'm going to approximate 61.5 to 62. 62 divided by 2 is 31. 31 is multiplied by log 10 to the 4. Now log has a base of 10, so these 10s cancel out, and our answer for this part is just 4. 31 times 4 is equal to 124 millivolts, and this value is very much positive. And it's positive because calcium has a positive charge, so that makes sense. The answer choice that's closest to this is going to be D. So 13 is D. We can now move on to problem 14. Problem 14 says, which of the following statements conflicts with the fluid mosaic model? So for this problem, let's work through each answer choice. A says the cell membrane is static in structure. The fluid mosaic model actually describes the membrane as fluid, meaning the lipids and proteins, they can move, and that creates a flexible and dynamic environment. If we call the membrane static, that would imply it's fixed and rigid, which directly goes against the model. So this one is incorrect. B says membrane components can be derived from multiple biomolecules. This statement is true. The membrane contains phospholipids, cholesterol, glycolipids, and proteins, all of which come from different types of biomolecules. So this is consistent with the fluid mosaic model. So far, statement A is false, statement B is true. Remember, we're trying to figure a statement that conflicts with the fluid mosaic model. We already kind of have an answer, but we're still going to go through C and D. C says hydrophobic interactions stabilize the lipid bilayer. This is also true. The nonpolar tails of the phospholipids, they interact with each other to avoid water, and those hydrophobic interactions are what keep the bilayer intact. Finally, answer choice D says proteins are asymmetrically distributed within the cell membrane. Also true and correct. So certain proteins, they can face the extracellular space while others face the cytoplasm, and that asymmetry is essential for cell function. So reviewing all of these, the only statement that actually conflicts with the fluid mosaic model is A, because the membrane is not static, it's fluid and dynamic. 14 is A. With that, we have our last and final problem. Problem 14 asks, which of the following is a sphingolipid? Now, from lecture, we learned that sphingolipids include several subtypes, such as ceramide, sphingomyelin, glycosphingolipids, and gangliosides. And looking at the answer choices, the only one that matches this category is D, ganglioside. So 15 is D. Very straightforward question. Lovely one to end this problem set on. I hope this has been helpful. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.